Okay, today we're talking about microarray background correction. We're going to focus on the Affymetrix platform, although some of the issues we're talking about today apply to other platforms as well. Alright, well, we start with an MA plot, which we explained in the useful plots lecture earlier. And this is a MA plot comparing two samples that are basically technical replicates. With the exception of 16 genes, these are technical replicates. So in principle, all these points should be at 0. There should be no differences, but we see hundreds of points outside of the fold change 1 region. So that's a problem, and they all happen to be at the problem is worse towards the low expressed genes, right? Remember, A is measuring, quantifying how expressed is the gene on average between the two samples. So we see a bigger amount of false positives for the low expressed genes, and today we're going to understand why this happens. It has to do with the way the raw data is processed. So to understand that, we have to go back to the original raw data, and the explanation is going to be specific to the Affymetrix array, which is illustrated with this cartoon here. The way it works, this is one gene, and the cartoon is showing you the reference sequence with this line, and the blue little lines are showing us where the probes are. So the probes, we use about 11 probes to identify, to represent one gene, and they're towards the 3' of the gene. That's done for technical reasons having to do with degradation. So we have 11 probes. Each one is 25 bases long. Here's an example, and this one, we say it's this probe here, this feature here is this probe, and it's this probe here representing this part of the gene. This is a sequence that will perfectly match. That's where the PM comes from, the reference sequence. All right, and we'll have 11 different ones, and then we'll take an average. We'll talk about that later in another lecture, talking about how we average these values. Now, one observation that was made early on was that there was background noise. This comes from cross-hybridization. This comes from the scanner optical noise. This comes from other factors. And what was difficult, what made it particularly difficult, is that the background noise levels were probe specific. So every probe appeared to have different amounts of background noise. So Affymetrix designed a probe just to measure the background noise. So for each probe, for each PM probe, there was a mismatch probe, an MM probe, paired up. They were identical except for one base, right? So the idea is that by changing this base, this probe will not match, will not hybridize to the signal we're trying to measure, to the transcripts we're trying to measure, but it will bind to all the other stuff that is also hybridizing to this probe by accident. So then this will serve as a form to correct this measurement. The measurement coming from the MM will be useful for correcting the PM intensity. That's the idea. Okay, so before I continue, I want to explain the data, an experiment that I'm going to use to show, a couple of experiments actually that I used to make several points. So I want to explain the experiment first. So this is it's called the Spikein experiment, and basically what was done, there have been various Spikein experiments. The one I'm showing you today is one performed by Affymetrix, and what they did was they took a number of replicate arrays. In one of these experiments there were as many as 60 arrays, and they were all replicates. So if you compare them to each other, they should give you the same answers. But they took 16 genes, 16 probe sets that were known not to be in the sample, and then they spiked in quantities for each one, so that we knew how much there was, right? So this was done in the lab, and the data is actually available in a package from Bioconductor if you want to download it and look at this data called the Spikein data package. Okay, so it's very very useful because the fact that they're mainly replicates gives us a way to decipher how precise this technology is, but the Spikeins are going to let us see how well we can distinguish differences when they're real, or as I'm going to call it in this lecture, accuracy. All right, so this is the design of the Spikein experiment. It's based on what is called in statistics a Latin square design. So every array, here are the array types A through T, and then we have probe set 1 through 16. So the idea is that we spike in different quantities, each probe set in each array. So in array A, we don't add any of probe set 1, but then in array B, we do add 0.25 picomolar, then 0.5, and then we double each time. Right, so now we do that for the second probe set, but now we start at 0.25, and we double each time. So the nice thing about this is that if we compare two arrays, say A and B, then we'll have a series of genes. 14 of the genes will have expected fold changes of 2. Right? So 4 compared to 2, 8 compared to 4, 16 compared to 8. We'll have two exceptions. This one, which is not a fold change of 2, it's a fold change of infinity, and this one. All right? So the other thing we can also do is we can compare probe sets to each other, right? So we can see if probe set 1 and probe set 2 they're actually increasing. So it's a very nice design. It's been very useful for development of methods for data analysis. Okay, so this is a Spikein experiment. That was the design for the Spikein experiment for the older chip. There's a new version, and it's very similar design, and it's also part of the Spikein package in R. So you can download that and see exactly what it's like and use it however you want. Okay, so now the other experiment I'm going to describe is one where I'm using where an experiment was designed simply to look at background effects. So the experiment was quite simple. We hybridized a chip with nothing in it, a chip with just water, a chip with RNA but no label, a chip with yeast DNA that we know should not hybridize to the human gene expression array. Because we know the yeast genome, we can actually remove any probe that has homology. And we also did just a poly-C and poly-G hybridized to an array. So at the end of the day, really the one that was most useful was the yeast DNA chip. But anyways, I'm going to show you all the data that we obtained from this. And here's a box plot of the intensities obtained from each one of these. So when you just put water on, you still get intensity. That's because they're optical noise. So you get a distribution ranging from 2 to the 5, that's 32, all the way up to 2 to the 10, which is 1024. So there's quite a bit of range, although most of the data is down here. There's not much variability. Most of the data is below 64. And but the point is that it's not zero. We do get intensities even when there's no label, even when there's no RNA. Now, when we have DNA that shouldn't hybridize, then we see how cross hybridization affects this. So this is much higher than these guys. So this is a cross hyb experiment that we're gonna use here and there throughout this lecture. Okay, so now let's get back to this picture. So why is there so much noise here? Now that we understand what the MMs are and what background is, we're gonna explain why this is. It's actually due to the way that background correction is performed. All right, so why do we adjust for background? Well, it has to do with the fact that we never have zeros, right? So there's always some kind of background effect that shifts. It seems to be additive and shifts the data upwards towards positive numbers. So this plot comes from real data, and what I've done is I've taken the Spikein experiment and I have averaged all the probes that had 0 spiked in, so no transcripts, and I average them, and I can see that they're around 100 and some. That's the intensity you see. This is not in the log scale. Then I took all the probes that were at a quarter, did the same thing, then a half, then 1, 2, 4, and 8, and you can see that there's a linear relationship. That's very important that we see that. If this was flat, then we would be in trouble. This technology would not have been useful. So the fact that this guy is not at 0 means that there's some kind of background effect. Now I'm gonna call that alpha in the next few slides. That alpha is gonna represent background noise, and then beta is gonna be the true concentration, which which here is increasing from 0 to 8. So I'm gonna show that later. I'm gonna be using these alpha and beta notation throughout. Okay, now if we look at the same plot in the log scale, it starts to show us why this is a problem. So notice that if we were going to compare the array genes that were spiked in at a quarter compared to genes that were spiked in at a half, that should be a fold change of 2. However, we see that on average there is barely no change. Right? There's barely no change. It goes from 7.7 to 7.75. Now for the higher values, then we do see a bigger change, right? So this is going from 2 to 3. That's a fold change of 2, and it's it goes from 8.2 to 8.7. Sorry, 8.3 to 8.7. So it's not exactly 1 as it should be, but it's getting there. So that has to do with background. So here we're seeing alpha 1. That's the true expression level. Alpha 2, true expression level for the second point. But then we're adding a constant. Now, if that constant is equal, which could be, if that constant is equal and much bigger than the alpha, then this is going to be this ratio is going to be close to 1, which is consistent with this line being flat. Now up here, because the alphas are much bigger than the beta, they dominate. I'm sorry, the alpha is the background, so I just switched that. So if the alpha is not as big as the beta, if the beta overwhelms the alpha, then we get something closer to what we actually want to see, right? Beta 2 divided by beta 1. Beta is the actual concentration. I incorrectly said alpha at the beginning of the slide. But yeah, so if the background's high, if alpha's high, then it dominates this ratio, and if the alphas are similar, then this ratio is going to be 1. Over here, if the beta, the real concentration is high, then it dominates this fraction, and we actually get the correct answer. So these two should be comparable. They should both be beta 1 divided by 2, beta 2. So we need a way to correct these alpha out so that this ratio is, instead of 1, it's actually what we want it to be. So basically what I'm saying is we want this to be a line and not a curve like this. It starts flat and then becomes a line. That's the motivation for background correction. The main motivation, the practical motivation. Okay, so here's a big problem. That plot I just showed was the average across all arrays, all genes. If I then plot if I make the same plot, but for each gene individually, actually this is not even each gene, this is each probe within a gene. So I take one gene and I show all the probes for that probe set. This is an older chip, so it's 16. You see this flat at the beginning and then line behavior for every probe, right? So it's flat because of background and then straight. Now what's tricky is that the background levels are very different. So this purple one has a bigger non-specific binding levels than this orange one. So if that's the case, that means that we need to correct each probe with a different background level, and that makes it difficult. So the way to say that mathematically is that each probe has its own alpha. Well, that's why the manufacturer decided to put the probe, right? I call it the direct measurement strategy, where you estimate the alpha by measuring it, and then you subtract PM minus MM, and hopefully that gives you the beta that you want. So you have PM is background plus the real signal, MM is just the background, PM minus MM is the real signal that you want, and now we're ready to take log ratios and get the log ratio for say probe 1 comparing sample 1 and sample 2. Okay, now let's this is the plot. When you use that strategy, this is the plot you get. This is the date. This is the outcome that you get. And so now we're going to see why it's happening. It's actually explained mathematically why we get this variance explosion. right? So this is what we just showed. This model isn't quite right, and the reason is it's not quite right is because this part of it is not exactly like this. It's not as deterministic as is shown. So here I'm showing the data from one of the yeast experiments, and I'm showing you PM versus MM. Okay, so PM versus MM. This is the yeast experiment, so there's no beta, there's no real signal. It's all background, it's all alpha. So this plot, if it were perfect, it would be alpha versus alpha, and we would see a straight line. But we don't see a straight line, we see some scatter around a straight line. Okay, so if that's the case, if we see scatter, then a better model than this. These two alpha are not the same. It's more like this. So we have the PM is alpha 1 plus beta, but MM is alpha 2. Alpha 1 and alpha 2 are correlated. I'm using the log here because that's where, you know, it looks nice and normal-ish. The correlation is about 0.7 based on this picture, but it's not 1. When that's the case, when this correlation is imperfect, we can show mathematically that if you're taking because eventually you're going to take the log for the log ratios, you can show that the variance of the log of this difference. By the way, I'm putting a little star there because we have to be careful in cases where the MMs are bigger than the PMs. We're going to get a negative number, so we have to be careful about that. In any case, if you do something like this, then the variance of this quantity, of this statistic, is going to be inversely proportional to the signal squared. Okay, so what does that mean? When the real signal, when the real expression is low, the variance is big and it grows with a square, and that is what we see in that picture, in the original picture, in this picture, right? As it gets lower, as the beta gets lower, the variance grows as it grows as 1 over beta. The standard deviation grows that way. So it looks a lot like what that model is predicting. Okay, so I'm going to show you with a simulation that this is in fact the case, and it also is going to help illustrate how background correction does in fact improve accuracy. So it gets rid of, on average, it gets rid of background, but it increases variance. So to show this, I'm gonna simulate data. I'm gonna simulate 2 arrays. They have no differences. I'm assuming they're technical replicates, and so I'm gonna generate the PMs and the MMs using that assumption, and then I'm going to correct with different versions of log PM minus k times MM. So the 2 extremes are going to be when k is 0, that means we're not background correcting at all. And then at the other end of the spectrum, we're going to have k equals 1, where we are background correcting very aggressively. We're subtracting the MM completely. Now then I'm going to show you several points in between so you can see how the variance increases. So I'm going to show you an MA plot. This is going to be a good way to see how the variance grows. But then we're also going to make a plot showing us the observed data. So the observed data being this compared to the known truth. I'm generating this with simulation so I know what it is. And then I'm going to show you various values of k. So here's k equals 0. So here's the MA plot. We don't see a lot of variance. There's no red points. That means there's no points with fold changes above 1. And here's the accuracy. So the accuracy, we can see that for high intensity genes, it's nice and right on the line. It's very accurate. But when there's low intensity genes, then the background takes over, and we get very biased results. See, this green line is very far from where it should be, which is the orange line. Now, as I start removing more background, notice that the green line got closer. It got less bias. This line got closer to the red line. But now we see a little bit of noise. See how these red points appeared? Now we get a half. So the accuracy gets better, precision gets worse. Then we go to 3/4. Again, the same thing happened. Accuracy got better, precision got worse. And then we go all the way to k equals 1, and we have quite, these results are pretty good. They're, on average, it's pretty accurate, but now we're getting a lot of noise. And this looks a lot like the real data. So this is now The original algorithm looks a lot like the simulation I just showed, and this is what you get using a background correction algorithm from the RMA algorithm, which we are going to explain now. And it's similar to not doing background correction. So this is what the RMA algorithm does for background correction. It uses a mathematical model to background correct. This has also been called the normal exponential background correction model. It's being used in other platforms, other technologies. So here's the idea. We're going to assume that the PM is alpha, that's the background, plus beta, that's the signal. We're going to assume the alpha is normal with some mean and some sigma, and this is going to be independent across probes. So it's an empirical Bayes model that assumes the distribution across all probes. So this is on one chip. This is not across chips. This is on one chip. So the beta is going to be assumed to be exponential with some value lambda. So the reason we're doing this is because now we can use an empirical Bayes approach that lets us, first of all, estimate mu, sigma, and alpha from the data. So you use the data to estimate these three parameters. And once we know those parameters, then we can estimate beta, we can predict beta given PM. And we do it in a way, using statistics, we estimate beta given the PM, and what this quantity minimizes is going to be the mean squared error. So instead of focusing on bias or precision specifically, it's going to minimize the mean squared error, which is a combination of both. Both variance and bias have to be reduced for this value to get for the mean squared error to get small. So if you do that, if you're using this assumption, then this has a nice closed form. This is one reason we chose these distributions, because we get a closed form. We don't have to do any kind of numerical approximation to the estimate of the signal. Okay, so one of the things I should point out here is that nowhere in this formula do we see the use of the MMs. There's no MMs. So one of the things that this did is it took away the need for the MMs if you trust this is a background correction method, and in a very practical downstream result of not using the MMs is that we can put twice as many probes on the chips, twice as many PMs, twice as many genes. Okay, so this is the original approach, the PM minus MM approach, and you can see now these greens, I can explain what they are. These, because this is a Spikein experiment, these are the genes that have a fold change of 2. That's what they should be. And then this one is the 1000 versus 0 gene, and this is the 0.25 versus 0 gene. So when I, instead of using this background correction approach, I use the RMA approach, then it turns into this. So you can see that obviously the background noise has been taken away. The signal is still there. We still can distinguish these genes from the noise. Although one negative is that this 0, this gene that was very different before, see, it was up here and should be different, that has been attenuated towards 0. So that's the trade-off. So we remove variance at the low end, but we also might make some real things slower. Now, I think this method is now preferred, and most people use it over the old one. And I think it's mainly because this is in the right location of the ROC curve in terms of accuracy and precision. Although you might lose a little bit of signal, the gains in precision are so strong that you end up with better results, better downstream results. Now, the fact that we don't use any probe-specific information for this background correction method makes us think that for sure we could do better, because we know that background noise is probe-specific. So that's when we started trying to develop a method that would take the probe information into account, and I'm going to explain that new procedure now. So what we do, originally did was try to find sequence, try to use the sequence of the probe to predict the background. I'm going to explain that in more detail now. So first of all, I want to show a plot of why there's a group led by Felix Naef that noticed there was a weird shape when you looked at PM versus MM. So if you look at this plot, this is the PM versus MM on a regular chip. You can see that there's like a claw. If you look closely, you can see there's like a group here and a group up here. Here's a close-up of the high intensity gene. So there's this group up here, and there's another group down here. So there's a pretty big group where the MM is larger than the PM. So it turns out that the sequence explains this bimodality. So if I look at just the probes that have a C or T in the middle, then I get this. So these guys are the ones where the PM is bigger than the MM. If I have A or G in the middle, then I get some of the other group. So there's more to it than this, but there was a lot of this was explained by sequence. Okay, so this means that the MMs are, you got to be careful about using the MMs because there's this sequence effect and it's very strong in the middle base. So it's something that we have to be careful about. All right, so this plot here is looks complicated, but it's not that complicated. What we're doing here is we're trying to see what are the effects of probe sequence on the background. So what we did is we fit this model to the yeast data, where we know that it's all background. So you fit, you take the log and you fit a model that is the following. The model says you fit a mean level at each location of the probe. So there's 25 locations because they're 20 bases long. You fit a model that says that for every base at every location will have a different effect. So this mu, there's going to be 100 of these mu's, but because of constraints, it's actually going to be 75. So the mu's are going to have, there's going to be mu for each sequence A, T, C, G, and then the K is for every location 1 through 25. So these points here are the mu hats, the estimated mu. Right? So you can see that the C's have the biggest positive effect and the A's have the biggest negative effect, and the effects are bigger for bases in the middle. So if you have a probe that has several C's in the middle and you have another probe with several A's in the middle, you can get differences, you can get fold changes as big as 2 just based on sequence, not having nothing to do with biology or anything else, just based on sequence. And that is something that we could try to do better at. So this is the general GCRMA model. It's quite complicated. You basically assume that the PM is optical noise plus hybridization noise plus real signal, but that the background noise depends on alpha, which is an affinity measure computed from the sequence. So now we have sequence in the model, and we actually could also implement MM. You can do it with or without the MMs. The MMs we assume also has a sequence effect. And then what you estimate is the expression quantity given that you observe PM and MM, and that's the solution to this this model, which is actually not closed form, is what the GCRMA algorithm does. So GCRMA is not only looking at GC content. That's something that's maybe a little confusing from the name. The original version was looking at GC content, but then after we saw Naef's paper, we changed it to using all bases. So what we minimize is this. This is the mean squared error we minimize. And now again, borrowing strength and using an empirical Bayes approach, we get a background corrected value for each probe. Now this is RMA, so I'm going to show you that how it helps us recover signal. So it's it's nice and tight. There's no points outside of the 1, and these guys are these guys that should be different are quite separated from the noise. But then these two guys are, and these 1, 2, 0, 0, those guys are a little bit problematic because they should be up high, but they're not, especially this guy. This one should be way high because that's 1024 to 0. That's the real fold change. So with GCRMA, we do get a recovery of signal. These zeros go way up, the 2 and the 1 go up, but we also get a little bit of added noise. So you see that some of these black points, it should be 0, also went up a little bit. So that's what GCRMA does. It can help you bring out some of the signal at the bottom. It's not as robust as RMA, but it does have that property. So some people prefer it because of that. Other people prefer the stability of RMA. Okay, so in some real data, I just want to show that this has all been based on the Spikein experiment. So I'm going to end the talk showing RMA versus mass5 on real data, and that it does have a real effect. This is a plot showing distance. We've talked about this in the distance lecture. This is a MDS plot, and you can see that the different tissues. These are 5 different tissues. Separate out pretty well. You want each tissue to be close to each other, but far from the others. Now, in this case, we do have these. See, these 2 placentas are kind of stuck into the colon. That might be due to noise, and then this colon is closer to the midbrain than it is to its fellow colons. So with RMA, some of these issues go away, and you get a more compact result. Okay, and that's the end of today's lecture.